start recording. Okay. So holding the cells to form tissues. Now we are discussing about the cell to the nearby outside, which is we call ECM or extracellular matrix. So as we see in this figure, this is a lipid bilayer, and downwards, these are inside the cell. So we call cell cytoplasm. And as Jibom showed very nicely about the actin filaments that can polymerize and change the cell shape and even allow cell migration. So there are abundant actin filaments, and that also is connected to one part of this membrane protein here. It's called integrin. Integrin binds the cells to the outside of the world, especially extracellular matrix here. So you can see there's collagen, elastin, and also fibronectin molecules. So ECM proteins have a role of one is structural function, and second is adhesive function, so attaching together. So in structural molecule, we already talked about collagen, which allows the strength of this tissue binding, and elastin for elasticity. You hear a lot from the cosmetics industry. You see like elastin, right? So, and also adhesive function. When you grow cells, you often hear that some people, hey, my cell doesn't attach to the flask. So I checked it and I found fibronectin has to be coated. So fibronectin is a cross-linker in extracellular matrix to collagen. Here, the collagen, or a GAG protein, which is glycosaminoglycan. So, and also binding sites for adhesion receptors on cell surface. So, and that's one kind of molecule, fibronectin. And also, if these cells are, let's say, epithelial cell, you're likely to also have heard about laminin protein. It's binding sites for cell attachment. Because in fact, you remember all epithelial cells are sitting on top of a basement membrane, and that basement membrane, the component is a laminin and collagen type 4. Okay, so that's why here fibronectin or laminin can be used as an example of ECM protein. So glycosaminoglycan and proteoglycan, I think I mentioned it before. And you hear when you go out for like shelf drugs store, you can find the GAG protein. It's good for your cartilage because this contains a lot of ECM molecules. So it provides a source, but there's not clear evidence that actually taking GAG will improve your knee function. When you have a diabetic patient, they may have to be careful about taking this too much. So what's the most abundant protein in our body? I think you probably already know now that's containing about a quarter or more of our whole protein is composed as a collagen. So I think here is, sorry, it's a collagen molecule. And what are the major components of basement membrane and produced by whom? As I already said, a basement membrane is composed of a laminin and collagen type 4. In fact, collagen have dozens of subtypes. So sometimes you have to, I only remember one. Collagen type 1 is fibrillar collagen, okay? So that's also very important. And collagen type 4 is forming the basement membrane. And then the question is, who make this basement membrane? Let me ask one of you guys. Let's see. 락스만?
PPT가 안 움직이는 것 같습니다.
안 움직여요? 지금은? 안 움직여요?
안 움직입니다.
Okay. Okay, sorry. Let me restart. So how about now? Let's see. Okay, can you see the PPT? Can you see the PPT now?
Yeah, we can see.
Okay. All right, so then I think I'll have to move it by my hand. So let's see. Yeah, so Laxman?
Yes, Professor.
Yes, can you guess who makes this basement membrane, which is laminin and collagen type 4?
Which cell? Sorry, Professor, I have no idea about that. Which
That's
cell?
fine. You don't have to be sorry. But remember, basement membrane came when there's epithelial cells, they will be always sitting on top of the basement membrane. What that means is actually epithelial cells secrete and make their own basement membrane. So this is what I want to tell. That's made by basement epithelial cell. Is it clear? Okay, so the question is, why are we talking about this ECM for this BME class? And that's something I want you to know, that let's say we are developing a tissue engineering. So artificial scaffold, remember, for tissue substrate, we provide scaffolds, and then we have to seed the cells to hoping that the cells will form an artificial, let's say, skin substrate. But to make this happen and good, the scaffold often need to be coated with this ECM molecule, such as fibronectin or even peptide called RGD. This is a single biochemistry like amino acid, so that R stands for arginine, glycine, aspartic acid. So this RGD is famous, that integrin binds to this RGD of the extracellular matrix. So that's why this is important to understand when you do tissue engineering and grow cell culture and then try to develop a tissue substrate. So that's something I want to share with you. And now let's move on to talk about membrane proteins. Last time I talked to you that the cell membranes are covered or embedded with a bunch of proteins and globular protein and many transporters, ion channels, and these are very, very important. And what kind of these membrane proteins are there? You can see these transporters. For example, like sodium and potassium. These ions are not normally can pass through this easily. So there's a special type of this transporters or ion channels. And also glucose molecule, even small, it's a 6 carbon sugar molecule, but the cells cannot just take it directly. It needs to have a glucose transporter, and then you can actually transport glucose. Also, a bunch of these proteins, such as integrin, they serve as a anchor protein. Also, very important one is you probably have heard about receptor molecule. So this receptor molecule is related to the signaling. What that means is, actually, I think this is a good picture to explain. So there are receptor and ligand. So ligand is a primary messenger. It could be like growth factor. It could be hormone. It could be kind of protein, which is supposed to be fit into the receptor. And then what it makes is it forms these molecular changes, which we call conformational change. And that triggers a downstream signaling. You can see this, we call multiple arrows. And this explains what's happening as it propagates that even smaller at the beginning, but it will activate many signaling molecules, such as like kinase molecule, which what it does is actually phosphorylate the downstream molecules, and it activates that enzyme or protein, and then they will propagate. Okay, so this cascade we call signal transduction, or this is second messengers, because there are ligand is primary messenger. And what does this eventually lead? It's first cellular responses, such as cell migrating towards the bacteria or something, and it also enters into nucleus. It can change gene expression, or it can allow the cell to divide. So this is such an important role that modern medicine, almost over 50% are targeting these membrane proteins. So you see the importance of these membrane proteins. Of course, there are also enzymes which does directly allows a reaction. So membrane proteins are attached to or associated with the membrane of a cell or organelle. And the function of membrane proteins, first transport of molecules such as glucose or ions. And one example is sodium potassium pump, which is, inside the cell. Outside of the cell, there are a bunch of more sodium molecules, and inside the cell, there are potassium molecules. And this pump actually works as an adverse gradient to keep the homeostasis. And this requires an external energy because this is not simple diffusion, which allows moving these ions against their original concentration gradient. And so it requires energy, and that energy is usually coming back from ATP. So they use ATP in this specific type of pump. And also, as I said, there are adhesive molecules here, like anchors. There are several types you hear, but I'm sure you will face this a lot on your career. So there are integrin molecules, which is responsible for the cell attaching to the outside, such as these collagen molecule outside. So this actually the cells sense mechanically whether they grab to whom and how, what kind of forces are there. And also the selectin molecule is used for interacting with the glycoprotein, which means a sugar protein of the immune cells. You remember that? White blood cells and red blood cells. The white blood cells are flowing along the blood vessel. When there's an inflammation, selectin molecule expressed on the endothelial cells. And these white blood cells actually interact and catch and know that there's something going on inside the tissue. So this kind of messaging, selectin is involved. And cadherin is, as a GBOM shows, excellent way of a cell-cell attachment and cell-cell communication. So cadherin molecule is part of, like these epithelial cells. Between epithelial cells, they attach, and there are like tight junction, and also this molecule called cadherin, which attach those cells. So these adhesion molecules are also known. And of course, the signaling, I think I just described you. So I want to show one kind of a diagram which looks like a map, which is called signal transduction pathways. So those who are working on more like cell biology aspects will see a lot of these. So why do I show it to you? Because I want to explain what's our understanding of what's going on inside the cell. We know a lot now, but we do not know everything. But let's see, these are the cells. And so these membrane proteins, for example, like this IGF-1, which is insulin like growth factor 1, which is growth factor. So that's a ligand, and this is providing survival. So when it interacts with a receptor called RTK, stands for receptor tyrosine kinase. Okay, kinase is an active enzyme which phosphorylates another protein, another kinase. So it activates PI3 kinase, then it will activate AKT and blah, blah, blah. So, and these eventually lead to gene regulation, okay? And also GPCR, a famous G protein coupled receptor. The discovery actually led to Nobel Prize in 2012. The reason is there are many, many drugs actually targeting this G protein coupled receptor, which is chemokine hormone transmitters interact, and then they eventually, you see, go to this Krebs cycle and gene regulation. Okay, so these are something I want you to understand why membrane proteins are so important for the cell to communicate this microenvironment. Also, at the same time, mechanical extracellular matrix interact with the integrins. Or what about this, the other one, death factor? There are like death signal, which you may heard from cell biology, like caspase-9 and this BAM-BAD BCL2. These signals, there are also signals for the cell, hey, you gotta die, apoptosis. Okay, or other like this growth factor eventually lead to cell proliferation. All right, so this is so complicated. And let's assume these cells are cancer cells. You don't want this cancer cell to keep proliferate. So you realize by growing the cell and then check out the amount of mRNA and the protein expression. For example, you've got a lot of a growth factor. Let's say this MAP kinase, you realize this is a lot. So, okay, I'm going to target this cancer cell. So you develop a drug called EGF, epidermal growth factor receptor blocker. So you've got blocked this one. And that's we call targeted drug. There are already like biotech companies made this drug. The issue is if you block this pathway, the cancer cells somehow develop another path to grow themselves. So this is the limitation of current anti-cancer drug. Many of this targeted approach for cancer treatment. Why do they fail so much? Because you can't target only one pathway. The cell somehow develop the other ways to survive themselves and keep proliferating. So this is something I want you to know. So what about the strategy for this? Okay, you give this drug, you find out this MAPK is an expression, high expression is low, but let's say you have find a PI3 kinase, this is becoming high. Okay, then I'm going to block this one too. So it's called combination therapy. And you hope that, hey, this will kill the cancer cell better. For example, drug A and drug B combination. You realize that combination therapy gives an additive or synergistic effect. But the problem is, these pathways are very important for normal cell growth and function. Sometimes you are increasing toxicity. Then you have to also see whether it's too toxic that you cannot use this drug, multiple drug with high dose. So there are balances for that. So to finish this part, I want to move on to cell cycle proliferation to differentiation and stem cells. Okay. Let me check. Okay, so can you see this well? Raise your hand. Okay, so let's continue about controlling cell cycle. How do cells know when to initiate DNA synthesis and start to divide, mitosis? And that decision-making factors can come from inside or outside, okay? Let's first think of inside. Cellular protein itself is a biological clock. Of course, when we were developing embryo, the cells are programmed to keep proliferating, okay? But once we become an adult, once our growth is up, still there are times we have to grow. That depends on biological clocks, special protein to promote and inhibit cell cycle. You remember about a cell cycle. And so, for example, your skin and your gut, okay, they have to continually renew. And scientists studied some specific type of molecule, like goes ups and downs together with the cell cycle. So they name it the protein as a cyclin. Okay, so there are CDK molecule called the cyclin-dependent kinase to allow this to happen. Okay, so what about the outside? So for the cells are, as I said, through the membrane protein, always communicate with their outside of the world. Especially there are growth factors. You are learning a lot from cancer, then you hear a lot about this EGF and VEGF, which is EGF is epidermal growth factor, VEGF is vascular endothelial growth factor, so famous for angiogenesis, okay? These can bind membrane receptor. What they do, trigger signaling cascade to eventually express growth promoting gene, and then they make the cell to keep divide, okay? Because it's growth factor. One thing I want to draw your attention here is a very interesting enzyme named here. So these DNA, the genome, at the, like the genome, you have to replicate. So at the genome, at the terminal site, there are like repeating sequence, and that is at the end of this genome. So people named it as a telomere, okay? It's at the end. So with the telomere responsible for overall lifespan and have role in cancer therapy. And in fact, this is very interesting that, once the cell divided 1 to 2, 2 to 4, and what happens is this genome at the end, it's continually taking off, okay? So this telomere keeps shorter and shorter. And once the cell divides a certain number of times, the telomere is run out and the cells undergo programmed cell death. So that's why when you do primary cell culture, you cannot keep the cell in your culture dish and passage it forever. But interestingly, the cancer cells, over 90% of cancer cells express an enzyme which keeps adding telomere at the end so that, immortal cancer cells can divide forever. I mean, that's very interesting thing because of this potential therapeutic effect. This discovery led to a Nobel Prize in 2009. Okay, what would be the name of this enzyme? Brahanu? Can you guess what this enzyme's name? I said
Polymerase.
Yes, great.
Polymerase.
Exactly. I said the enzyme usually named finish as ase, so it's called telomerase. So over 90% of cancers express telomerase, and that means blocking this might lead to anti-cancer therapy. So cell proliferation, I'm sure you've got this, learned this from your previous times. So I just want to point out when to divide and proliferate for the cell. So you've seen these cell cycles. Many times the cells are in the it's like static position, which we call G0. So this is a resting state. Okay. But when there are signaling which ask the cell to divide, and it starts to divide, that leads to the S phase, and this S is when interaction of cell with the environment by, for example, growth factor. Then what they have to do is they have to synthesize DNA. Okay, so DNA synthesis, that S we call S phase, and after this synthesis, you need to have a prepare not only DNA, but the single cell to become double, you need to have double the organelle and proteins. So you need to prepare for that. That's why we call G stands for actually gap. Between these major events, this is a gap phase. So G2 phase called gap 2. And then now afterwards, the cells can actually divide, which is mitosis phase. Okay, so that's called M phase, somatic cell division. Okay, and this mitosis is actually subdivided by prophase, metaphase, anaphase, telophase. So more details are in this YouTube. So I want you to watch this later on. Okay, so first growth phase, you need to grow the cell. So that's a gap 0, growth and normal metabolic, and then DNA replication, synthesis, so S phase. And then you have a gap before mitotic, you have to grow and preparation for mitosis. And then you have mitosis. Okay, and when this division happen is a question. So of course, when we are embryo, we are keep doing this because we have fastly grow. But now, like us, we are adults, right? When we've got in there, let's say a cut here, and then the cells, nearby cells, now get the signal, and we have to divide to fill up the loss part of our tissue. And not only this injury, but normal tissue function. For example, our vessels are covered by endothelial cells. there are a small percentage of cells keep replenishing themselves. Okay. Also, but that's endothelial cell. The more active ones are your mouth and GI tract, and these epitheliums are always facing, you know, foods, but also sometimes bad ones. So it keeps regenerating itself. So, for example, your gut, that epithelium is regenerating every like several days. So that's a normal tissue function. What about when we got COVID virus and our immune system got it and detect, or vaccine, and why we are like kind of having fever or something? Because we've got to generate a new antibody, which is produced by B cell. Then you have to produce the clonal expansion of that specific B cell, which produces antibody against this epitope or antigen molecule, which is the vaccine. Okay, at that time we have to keep proliferate. So that's immune response. So that's when we need the cell proliferation. Let's go back to the differentiation during the embryonic development before we talk about stem cells. So we have a diploid cell form two haploid gametes, okay, and that we call a zygote, okay. So zygote is a fertilized ovum, and that zygote, once they develop, they will form hollow organ, hollow cell, and that inside the inner cell mass is this is called blastocyst. And on the placenta, they will start to develop into a layered structure called ecto, meso, endo, meaning that external, middle, and internal layer. Their fate for development are a little different because ecto goes to skin or neuron or pigment, while the middle mesoderm becomes cardiac, skeletal, or kidney cell, RBC, smooth muscle, while the endo, the inner side, will become a lung cell, alveolar cell, thyroid, pancreatic. Of course, there are some cells which will become these germline cells. And so these mammalian blastula with some differentiation, with you see this small inner cell mass, that we call blastocyst. Okay. And because the cyst means a small sac. Okay. So again, the question, are these many different cells, are all DNA in different cells in our body the same or not? Let me ask this question to, let's say, Songhyun?
Yes.
Yeah. Can you answer this question?
Yes.
Of course, yes. The key part of this, you know, DNAs are the same, but the gene expression, different gene expression makes different type of the cell. And this terminology review, I'm gonna skip because I already talked a lot about this. So what I want to tell is this outer layer was a ectoderm, because ecto means outside, and meso and endoderm, so that's within, and haploid, diploid, a zygote, as you see, male gamete with the sperm and female gamete with n number of chromosomes. When they fertilize, that form the zygote, which is 2n, and this now will become a differentiating. So now I want to get into the stem cell part. So this is something very important and hot issue more recently about the cell differentiation and stem cells. So what is the cell differentiation? That is the process of cellular specialization in function during organism development and tissue repair and normal cell turnover with the same genetic material. So especially, I guess you all wonder, like, hey, when you are young, when you have a like injury or like skin abrasion, or you always see that regenerates. We wonder why this happens, what happens. And also your hair, for male, you have this hairs keep growing. Even if you do shaving, it keeps growing. Hey, what's going on up there? There comes this stem cell, okay? The stem cells are unspecialized precursor cells, okay, that can differentiate into diverse specialized cell types or can self-renew. In developing embryo, stem cells can differentiate into all the specialized cells, and that's the power and interest of stem cell. And in adult organisms like us, and stem cells, as I discussed right before, act as a repair system for the body, also maintain normal turnover of regenerative organs such as blood, skin, or intestinal tissue. So for example, you know, our red blood cells, can you guess how long the red blood cell can live, survive in our body?
3 to 4 days.
Oh, good guess. And in fact, let me see. Oops. Let me see. Where are they? Yes. In fact, the red blood cells can live about 120 days. And you know, the importance of the RBC function is, you know, our capillary is very narrow. The RBCs have to be very flexible to pass through these capillaries and to exchange oxygen outside. But as time goes on, the RBCs will be stiffened and they got caught in your spleen, okay? And then they will disintegrate over there. So there gotta be something which will keep generating your red blood cells, okay? So that's where stem cells involved. In fact, that happens by the cells inside your bone marrow, okay? So where are these stem cells? It's a real question. Important thing is all multicellular organisms, they all have stem cells, okay? For repair and for self-renew, okay? So then we have to ask us, why are we talking about stem cells in biomedical engineering? Yeah, their potential use in repopulating and tissue. Let me ask. Kuhinor?
Yeah.
Yeah, can you guess?
Regeneration.
Yes, exactly. Yeah, tissue regeneration. So for biomedical engineers like us, it's very, very much of our interest, okay? Because we are interested in injured or lost or malfunctioning or diseased organ can we replace? Okay, that's a big question. Or from bone injury, can we replace our skin? So then we have to look for our natural ways of repopulating and healing and regeneration, and that's coming from stem cells. So stem cells are potential source for cell replacement therapy for disease, also for engineered tissues such as skin replacement for bone injury, and like if we want to seed cell, we better to seed stem cell. Okay. So also, another point of concept of stem cell, I want to give you an example is in cancer. So in cancer field, we have like this multiply proliferating tumors forming, and then we give like anti-cancer drug, for example, chemotherapy to kill whatever which is dividing, so that we can kill majority of the tumor cell mass. But what if tumors also have cancer stem cell, which may be a few, but they are having the potency to quickly regrow to form tumor again. So this is the concept of stem cell, cancer stem cell field. Although this is so complicated, we still do not understand clearly yet, but that's the concept. You gotta kill and find out those stem cells. If your target is killing cancer cell, then check out the cancer stem cell. That's the approach and concept to figure out what are the characteristics of stem cell, cancer stem cell in cancer, for example. So then let's talk more about, so what are the stem cells? We need to understand the definition of stem cell first. So as we discussed, in vitro fertilized egg, and that egg, which is coming from sperm and egg sexual reproduction, and that fertilized egg can become the whole organism. So this has the potential to become everything in your body. So that's why we name it as a totipotent. Toti means it's from total, right? So that's from a Latin whole. And then it starts to divide. 1 become 2, 2 become 4, and become 8. So 8-cell embryo, and that will become a small inner cell mass of this blastula. And we call this as a blastocyst. All right. And this blastocyst, when you take it out and onto your petri dish, yeah, this is a stem cell, and this can become almost any part of your body, such as neuron, cardiac tissue, or connective tissue. So because of that, we name it as a, hey, this can be a multiple things. So we name it as a pluripotent. And that pluri comes from very many. What it means is, English, singular versus plural. So that pluripotent cells. And they become, after differentiation, they'll become a stem cell, but that stem cell is a little bit more specialized. Hey, I'm going to be only become muscle cells. I'm going to become only for blood cells. And at that stage of stem cell, we call this become only one. So that's why we name it as unipotent. Okay, hopefully this makes sense for you, because the potency or potent is becoming a power. So let me go over with you again that the potency definition. So can construct a complete viable organism such as fertilized egg. That is, in other words, the name is omnipotent. Okay, omnipotent, and that name is Yeong-Chan.
Yeah, totipotent.
Right, exactly. That's a totipotent. And then, as I said, descendants of totipotent that can differentiate to nearly all cells. Why I put nearly all cells? You just saw embryonic stem cell, okay? And that I said it's become plural, so it's a pluripotent stem cell. Then what are the difference between omnipotent and pluripotent? Euiheon Chung and others, and the differences, and these pluripotent cells are actually sitting on your placenta, on the mom's placenta. They cannot become placenta. Okay, that's why, except placenta, they can become nearly all. So that's why their name is a pluripotent. And then I also define new things for you as a multipotent stem cells, which can differentiate into a number of cells, but only those closely related family of cells. Okay, for example, we talk about blood cells. There are white blood cell, red blood cell, and platelet. So there are stem cells which can become blood cells, and that we call hematopoietic stem cell. And that is an example of multipotent stem cell. And of course, I said only one type, such as muscle stem cell, and these are unipotent stem cell. Okay. So now that's a definition based on potency, but based on their types of stem cell, there are embryonic stem cell, which I said isolated from an embryo. So a fertilized egg, you see, depending on some religious point of view, the way we are creating stem cell can become ethically problematic, because once it depends on when we take this as a human being. So once it's fertilized, some religious point, this can become a small human. So we are destroying it. So that can be the problem. This is we call embryonic stem cell, which is isolated from an embryo, okay, which is the most potent stem cell. Okay, so, but on the other hand, adult cells, adult stem cell exists. That's like us, adult organism. Let's say we take out some part of our skin cells, and there's no ethical issue at all for this. Okay, so people start to looking at an alternative which do not violate, do not affect the ethical issue. So from tissue of an adult organism, such as bone marrow, peripheral blood, cord blood, and those stem cells coming from this is called adult stem cell. And now, like a decade ago, even there are new discoveries called IPS. What it means is induced pluripotent stem cell. So we just learned the pluripotent stem cell, right? It can become almost all cells, and they are artificially derived from a non-pluripotent cell, typically an adult body cell, such as, let's say, your skin, okay? So this is body cell, adult somatic cell, okay? And this is called induced pluripotent. Why I discuss with you is, this discovery is what Shinya Yamanaka from Japan, his idea is, hey, can we take out already differentiated cell? Can we put it back to the stem cell stage by devising clever ways of giving different, like 4 transcription factors to reprogram the cell to show that this having a stem cell like pluripotent stem cell. And so this is induced by transcription factors, go back from somatic cell into pluripotent stem cell. And that paper is in 2006 in Cell. And you see, it's a surprise that only in 6 years, they got Nobel Prize for this. John Gurdon already made this, long time ago, a little, not exactly IPS, but this foundation. So what the Nobel Prize committee said for their discovery, that mature cells can be reprogrammed to become pluripotent by induced pluripotent stem cell. So this, become a hot issue, whether can we make this stem cell without any problematic embryonic stem cell or ethical issue. And so this is still, I think, in an active area of research. Then you can ask, okay, so I got an injury in my skin, okay? So it depends, the skins have epidermis, dermis, okay? So you know that how shallow, how deep your injury, it becomes a scar or no scar, okay? So when you've got a deep cut that goes deeper than the epidermis, you realize you got a scar and it may not regenerate well. So what indicates is there gotta be a specific location of our stem cell which can reproduce. But what if we take out that stem cell, they may not do their function very well. And these stem cells, our question is, where are the stem cells which will exist in usually small quantities within tissues of all our organs? Okay, so then how does this, I will get back to this again when I discuss stem cell niche. Okay, let's talk about stem cell differentiation. So what's interesting about the stem cell is their differentiation is quite interesting. Normal cells divide, 1 become 2, 2 become 4, exactly the same cell, but stem cells are different. Because look at that, stem cell divide 1 to 2, but somehow stem cell has to be there too. So somehow they divide one cells, when divide, it becomes self-renewal, becomes stem cell. It remains as a stem cell, while the rest is now become progenitor cells. Okay, what do I mean by progenitor? Okay, progenitor cell, which is a cell which has a tendency to differentiate into a specific type of cell and push it to differentiate into its target cell, ultimately the target cell. So that's progenitor cell. So stem cell division is somehow asymmetric, that, you know, the characteristic or unique property of stem cell differentiation, when it divides, it's symmetry one becomes self-renew. So, and that self-renew has to be fundamental to keep the stem cells there. So that is a limitless self-renewal. Okay? So that is a very important aspect of the stem cell. Okay. And what about the rest, which become a committed progenitor cell? They will start now to differentiate and keep differentiating. So we call amplifying division or transient amplification. In a second. Because of the monitor, I have to give the power to the laptop. Okay. Yeah. Sorry for the interruption. So this has to be multi-layered or multi-lineage differentiation to become a specialized cell. And because of this, you know, like exponential amplification, or we call this transient amplification, and it becomes a final differentiated cell, so that this can repopulate the tissue in the recipient. So in a nutshell, when you are talking, teaching your like friends or your parents, what are stem cells? You can say self-renewal and its potency to become any specialized cell. So it might be better and easier to just, it's easier, just talk about a good example. So let's think about our blood cell, okay? So the name's a little complicated. Hematopoietic stem cell. What the hemato means a blood, okay? Poiesis means to make. So blood making stem cell is easy. So think about the blood cells, and that hematopoietic stem cell can become multiple subtypes. For example, there are lymphoid, and other is a myeloid, and there are other erythro and megakaryoblast. So lymphoid is, you know, lymph node, and that lymph node, and also we have an organ called thymus, which is usually in your chest, and that's where these like immune cells are mature. So lymphoid stem cell become lymphoid blast, and eventually become B cell and T cell. And this immune cell we also call, because of the lymphoid nature, we call lymphocyte. Or because they are white blood cell, with the jargon, we call it B leukocytes as well. These are leukocytes as well. So these B cells mature in the bone marrow. Okay. This T cell matures in thymus. You see this T, and B is actually relevant. So B cells are producing antibodies. T cells are immune cells, which has like a self-cytotoxic T cell, or there are a bunch of subtypes. So these are white blood cells. Another type of stem cell is myeloid stem cell and the myeloid blast, and it differentiates into monocytes and granulocytes. So monocytes are circulating inside our blood, and once there's inflammation or when necessary, it comes out of the blood vessel to get into the tissue, and it becomes macrophage. You know, macrophage, the name itself says macro is big, phage means eating. So it's a big eater cell. So, you know, there's like invaders like bacteria, a virus, monocytes get into the tissue, become macrophage and eat up as an inflammation and function. And the other type of myeloid are granulocyte. You see there inside, there are, the even nucleus looks like lobular, and there are granules. They sometimes have to kill the nearby cells or intruder. So other types is erythroblast. Erythro means red, so it's erythroblast is become a red blood cells. And also megakaryoblast will actually pinch off, because it's mega, it's big, and it pinch off itself to create platelet. And interesting things, RBC and platelets has no nucleus, so they are not going to divide anymore. Okay, so this is all coming from hematopoietic stem cell. This is an example for you. So hematopoietic stem cell, in the end, it becomes multiple types. So let me ask question for Shukla. Is Shukla there? Yeah. Can you answer now? Hematopoietic stem cell is what kind of stem cell? Yeah. Can you guess? It's a multipotent.
Multipotent.
Yes. Yeah, the reason is, you see, stem cell can differentiate into a number of cells, but those closely related family, in this case, family of blood cell. So hopefully this will clear to you guys. And also, now let me continue the question of, hey, how many and where are the stem cells in our body? That's a still ongoing research, and I think for the stem cell engineering, for those who are tissue engineers who are dealing with stem cell, should ask this question to themselves, because we talk about adult stem cell and where are they in the body, and there are very interesting concept of stem cell niche. So let's see. There are many types of cells in our body and many different types of tissue. They require constant turnover. For example, hematopoietic system. Our blood cells, they all have their own lifetime, so we have to keep replenishing them. And intestine as well. For example, you remember like our small intestine, large intestine. Let's say small intestine has this villi, and they are epithelial, single columnar cells, you see, because they have to collect the food amino acid molecules. So there's only single layer, but they are columnar. And at the pit of this, like let's say these are like these small intestine, at the pit, there resides their stem cell, and they keep generating epithelial cells, and they push them into the tip, and the tip they'll fluff off. Okay, so their lifetime is only a couple of days. That's what's happening in our intestine. Endofollicular, this is for hair, hair follicle, and tissue with a low turnover. So there are some cells which doesn't seem to divide much. They are brain cells and our skeletal muscle. So our interest in this constant turnover, hematopoietic system, where are our old blood cells made? That's in the bone marrow. And I would say, so in the bone marrow, these hematopoietic cells can be made. Then can you just take out this hematopoietic stem cell, and on the Petri dish, they will automatically generate this RBC white blood cell? The answer is no. Our Petri dish is such an artificial place that, bone marrow provide a specific microenvironment that allow this hematopoietic stem cell becoming into different types of cells. Okay. And so that concept of this proper microenvironment is called stem cell niche. So another example, when you hear about immunology, B cell, let's say B cells are antibody producing cells, they have to have a differentiate from this original B cell, but they have to become specialized, specific, and their property has to evolve not everywhere in a proper place of bone marrow. And T cell has to mature inside the thymus. So when you engineer, I want to generate the T cell, which we can use it for biomedical or like treating disease, then you have to think about how the T cells actually from this progenitor to become mature T cell in the thymus. And that microenvironment, you have to understand there are biochemical cues, maybe even mechanical cues to allow the T cell from the immature to become mature T cell. So this concept was raised as we call stem cell niche, meaning that the microenvironment within the specific anatomic location where stem cells are found, and that interacts with the stem cell to regulate that stem cell fate to become which type of cell. So all this, why are we interested in stem cell? Because it can be useful for like stroke, traumatic injury, and all this disease may damage the cell. We need to replenish and we need to repair the system, or even missing teeth, wound. Okay, and spinal cord injury, many injuries, arthritis and blindness. Many of these stem cell, when we understand better, we may be able to treat this, our understanding and engineering of stem cell. Okay, so that part is for stem cell, and I want to move on to next, which is, we talk about the cell survival now. I'm talking about cell death. So one question is, when we develop from a like embryo to baby and think about our fingers, do you see the fingers when we grow? Does the finger cells grow like this? Or they're like just make a chunk and then kill the cells within and to make a digit? That's a question. And the answer is the second. So this is related to cell death. For example, this embryo, actually they make this and you see this red part, and they have a precise programmed cell death happens to make this red part keep dead, die, and then make viable these digits. So that's a fascinating aspect of utilizing cell death to make it precise of our organs, okay? And here, the question, I made this title as, hey, does the cell when die, they do suicide or homicide, meaning that self-death or the others kill the cell? And the answer is probably both. So the way of cell death is first, this is when the cell die, as you see the cells can dehydrate, shrink, and the chromatin become condensate, and you see this blebs are forming, and this inside the cell there's an active process of death, and we call these as apoptotic bodies. Okay, the important aspect is this membrane is still intact. Okay, so what does that mean? This means there's no big effect on the nearby neighborhood. And so this become recognized by big eater cell, macrophage, or even nearby cells. Look at this and then eat up. So it's a very clean ways of death, and that we call apoptosis. On the other hand, when you got injured, let's say you've got hit, okay, and then you see a bruise forming, and also like you've got toxins, like you've got maybe a bee sting, and then you start to see your tissue start to die. Okay, that abrupt phenomenon is a little different. So the cell and mitochondria swell, and then the plasma will rupture. And what will happen in this? Then the internal content, which is supposed to be inside the cell, comes outside. That will cause inflammation. the body will feel this, hey, something's happening here. Immune cells coming in, and what happens is you remember when you are hit by something, it will become reddish, which indicates that the vasodilation, your blood vessels will expand, and also the tissue will swell, which means this edema, which blood plasma can come out and tissue becomes swollen, and also pain. And all these are inflammation causing inflammatory response. And that we call as necrosis. So these two different aspects of cell death will eventually, you will see about how our tissue can be maintained is, the cells will proliferate and minus the normal or a cell death, and this will maintain the tissue. So apoptosis, actually in 2000, the discovery of this apoptotic pathway got Nobel Prize, and this is called programmed cell death or cell suicide. So why? Because it's really sequential, precisely regulated way of cell death. Chromatin condensation, cell shrinkage, and pinching off plasma membrane, forming apoptotic bodies. And this remains no or little inflammatory response. So it's clean death. While necrosis, it's like cell murder, okay? It forms a fatal cell injury. It's from fatal cell injury. So it's like unexpected, okay? So it's a premature death of cells and living tissue due to injuries such as trauma, or even disease such as infection or even stroke, okay? Blockage of blood vessel, it gives an ischemia, so the downstream tissue start to die, and that causes necrosis. In this necrosis, the cells swell and burst, releasing their contents, often trigger inflammatory response. And to help you understand further, I brought this a little more details, so please read through this for normal cell become a necrosis or apoptosis. The technology now, and the right side is actually my friend who built this microscope, which is called quantitative phase microscope, which gives a 3D, but the contrast is only coming from refractive index, so no exogenous fluorescent molecule or dyes involved. So we observe a live three-dimensional ways of how the cell death. You can see this blobbing, blah, 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 and this apoptosis, forming apoptosis bodies as a movie. So the technology allows us to even study this better. Okay, so now the rest is cell culture techniques, and this I will be a little fast. There are people who already do routine cell culture. There are people who have never done, and I hope in our department, whenever you have a chance, try this out, cell culture technique. Why in BME? Because our medical engineering, a lot depends on the study of the cells and tissues and growing this outside of the body. And that technique has been providing a good insight for our understanding. So that's cell culture and equipment. And I want to discuss about cell lines and primary cells and the references here. So cell culture is growing, maintaining cells under carefully controlled conditions outside of a living body, okay? And in vitro, we already discussed. Important thing is, in vitro experiments allows experiments that's very difficult or impossible in intact organisms. For example, I already showed multiple well plate that allow us to do parallel multiple assay with the same number of cells, so that we can have first row as a control, second row drug A, third row drug B, fourth row drug A plus B combination, and see how these cells will proliferate or die from this addition of the drug. That cannot be done easily in vivo, okay? So the goal of our in vitro experiments is really generating hypotheses about what occurs in vivo. You know, in vitro has this practical easiness, but you have to keep thinking the limitation, because the goal of this in vitro experiment eventually is to understand what's happening in vivo. Okay, so that requires careful experimental design and your interpretation. Okay, so always I ask you to think of, does the cells in a culture flask truly behave like they would do inside the body in vivo? There's always limitation, and you have to be aware of that. Also, for example, those who may be doing, let's say, cancer research, a lot of times we use cancer cell lines. For example, you know, recently we've got in mass media that about HeLa cell. HeLa comes from Heli Lux, a female name who died from cervical cancer. But that cancer was so aggressive that when the doctors isolate their cells, and they can grow well in plastic dish. So that has happened already 70 plus years ago. We are still using that to study, let's say, cancer. Let's say I want to test cancer treatment with this new drug, with this cell line, HeLa cell line, let's say. And that's immortalized cell line. This is so weird cell line. Are this a good model for real treating cervical cancer, let's say? You have to understand this one cell line coming from one single patient. We are dealing with the millions and thousands of real cervical cancer cell patients. You see the limitation. These cells are very weird, which is aggressive, which survives and transforms itself. So that's why in vitro, many people ask, hey, you got to use multiple different cell lines to prove or convince better. Even in that case, that doesn't guarantee that this will be truly killing and the tumor for the cervical cancer patients. So that's something you have to ask your question. Of course, immortal cell lines are easy to study, but there are logical gap and limit. So for that aspect, I want to discuss about the differences between and comparison between immortalized cell line, which, for example, cancer, there are dozens. For example, breast cancer cell line, there are dozens of breast cancer cell line, but there are millions of breast cancer cell patients. We are considering using this to treat the other, probably much more variable real breast tumor. Okay, cell line, why do we call line? Is the cell have been manipulated. Somehow they change to proliferate indefinitely. Okay, cancer cell line, right? Thus, but that's great because we can use it for long periods of time. While, hey, I'm interested in realistic. So sometimes some cell cannot become cell line. So in that case, we have to use the primary cell culture. For example, for those who are working on neuroscience, the neurons, you cannot grow them forever, okay? So the cells has to be removed directly from a real brain of a living animal or a central nervous system. So that's the difference. And you will hear a lot when you are studying reading literature about cell lines. 3T3, this is fibroblast cell line, HeLa, so I just talked, human epithelial cell, which is actually cervical carcinoma, HeLa cell. COS is a monkey kidney cell. PEC cell you see a lot about here, which is human embryonic kidney. MDCK, dog kidney epithelial cell. CHO cell is Chinese hamster ovum. You will see a lot because there are only a number of cell lines people are using, okay? So these cells are, of course, they are immortalized somehow. So they grow quick, easy to culture, great. And they are relatively well-known, relatively homogeneous. And you know, the reason why we study this multiple lab over the world is we want to compare each other. For that, immortal cell lines are great because they are already well characterized. And also, because you know they keep going, you can manipulate them expressing gene of interest. For example, like you have breast cancer cell line, you can insert, let's say, HER2 genes so that you can make HER2 overly expressing cell, or you are interested to delete them to see what changes are happening. So that purpose, immortal cell line is great. But you have to think they may not be relevant, okay? And also, because of this long time, you have to be very careful not assuming there will be exactly the same, because these too many passages may change the characteristic of cell. So you have to write down the passage number, and sometimes you have to maybe throw out and then get the new original cell line from the company, such as ATCC, which provides this cell line. While when you do primary cell culture, and of course, this is a relevant cell type, relevant physiology. And the results can be more confidently extrapolated to intact living system. That's great. However, you can't grow them forever. So limited lifespan. And when you take out, let's say, neurons, you take out the nervous system, you will not expect only neurons will grow. There will be another type of cells mixed together. So heterogeneous population with high variability will make you into trouble to make a clean experimental results. Okay. So now the question is, are stem cells close immortal cell lines or primary cell? Due to the timing, I'm going to just answer myself. Stem cells are coming from a living body usually. So in that aspect, it's like a primary cell. But stem cells have limitless self-renewal, so that it actually is almost immortal. So in that aspect, it has both aspects. In fact, the way people prove stem cell, hey, I isolate stem cell, is actually put it into the animal and check whether they form a tumor we call teratoma. Hey, I prove this is stem cell. Okay. And making this is you can dissect them. You can have a slice for organ culture, or fine chopping and having still multiple cells. It's an explant. And sometimes you use enzyme to isolate single cell to form a cell culture. And for those who do this cell culture, you see the cells will exponentially grow because I put the log number of the cell. And that we call, when you grow the cell and after when it becomes growing, you actually subculture them and then regrow. So this is how you propagate and subculture the cells. All right, so now 10:23. Let me only explain here and I'll stop. So x-axis passage number, y-axis cumulative number of cells. But you've got to be careful. The cells usually when they grow, when they hit each other, they stop growing. It's called contact inhibition. So it may not be exactly exponential. And also the surface property matters and the cell density matters. When it goes through and become forever, we call, hey, they transform, they become cell line. But normal primary cells, even cancers, you will see they become not growing and senescence. Okay, here I want to stop my lecture and I'll give you several minutes to write down your questions into the base camp.